Hello, my name is Alex Satz, and I'm going to be talking to you about the Dell screening platform. So let me jump right in. First, let me talk a little bit about how ardells are actually made. So let's say we have a mixture of three different things. We have structures shown here and barcodes shown here, and they're connected by a flexible tether. So we have a mixture of three different things, and we're going to split these up into three different tubes. So in each one of these tubes, we have a mixture of these same three things. And then in each one of these new tubes, we're going to add a new synthon, which creates a new structure, which is shown here. And we're going to add another DNA tag, which extends the DNA barcode as shown here. And in this manner, we are going to create 9 new structures, and we're going to create 9 new barcodes. After this step is done, so we've had a split size of 3, we've created 9 new structures. We can pool those 9 new structures inside of a single tube. And at any point in time, we can sequence off what the barcodes are. And from the sequence of that code, we will know what the structure is that is attached to that barcode. And so this is a single split size or a single step of split and pool combi-chem. And this is how we in Dell make a lot of chemical structures in a very short amount of time and at a relatively low cost. Of course, we don't use split sizes of 3. We use split sizes of 96 or even split sizes of 9,000. And so that we can create in this manner millions of new structures that we can screen. So thus far, we have synthesized over 280 different unique dells, which means we have created over 65 billion plus structures, which can be readily screened against a target of your choice. So how do you access these dells and these novel structures to be able to screen them? Well, we offer you 3 different ways that this can be done. So in the first case, you send your target to us and we screen it for you. And we can screen either a collection of 150 dells, or we can screen our covalent dells, which has 50 plus dells in it. An alternative to that is if you are an academic lab, we can send you a Dell Open Kit that contains 30 of these dells for free. And you can screen your target in your own lab, and then you send the results back to us and we tell you what your hits were. Or if you are a startup, or if you don't want to send your target to us at Wuxi because you want to keep the name of your target a secret, you can order a Delight Kit, and that contains 58 different dells. We can send those dells to you. Again, you screen them in your own lab, and then you send the results back to us, and we then do the rest of the work and share the results with you. So how does this workflow work? Well, first, how does the Dell screen work? So the Dell screen works by taking the target of your choice and immobilizing it onto a resin or a bead. You then incubate the Dell that we provide to you with the target. And most library members will not have a ligand that can bind to your target. And so all of these library members that don't bind are washed away. And what we're left with are the members that do bind to your protein target. And we collect these and we sequence the barcode. And from the sequence of the code, we know what the structure was that led to the binding. And then we share those structures with you, and those are the actual hits. So the workflow for this is always the same, whether you send the target to us or we send a kit to you and you run the screen in your own lab. The difference is, of course, this first step. Is the screening done by us or is the screening done by you? But regardless, the wet lab samples from that screen are then sent to us at Wuxi, and we do the rest of the work to determine what the structures of these hits were. And finally, we at Wuxi, we have screened over 184 different targets. And so the question is often asked what to expect from these screens. And of course, what kinds of targets do we find are successful in Dell screens? So I'm providing you here a list of different target classes that we have screened thus far. And shown in dark blue are the number of times that we have found a confirmed hit against that target. Shown in light blue are the programs which are ongoing right now. And shown in this light gray are the programs which have closed and which we weren't able to find a hit for. So not against every target do we actually find a confirmed hit, but against 95% of these targets we actually do. So I'd like to thank you for your time. And with that, I will hand it over back to Chris. Thank you.